Assalamualaikum warahmatullahi wabarakatuh. Hello everyone, we are from Group B and we will be presenting about roles of lower eukaryotic cells in pharmacy. So what is meant by eukaryotic cells? Basically, there are two types of cells, prokaryotic cells and eukaryotic cells. What are the differences between these two cells? In prokaryotic cells, they lack a nucleus, but in eukaryotic cells, they have a nucleus. Also, in eukaryotic cell, they have what is called an organelles, such as mitochondria, Golgi body, and chloroplasts. And we'll be talking more about eukaryotic cells. There are two types of eukaryotic cells, higher eukaryotic cells and lower eukaryotic cells. Examples of higher eukaryotic cells are plant and animal cells. Examples of lower eukaryotic cells are fungi and protists. Those examples are products of metabolism from microorganisms that are not essential to survival, which is called a secondary metabolism.
Now, I'm going to explain about the roles of protists in pharmacy. But before we get into that, I'm going to explain about the protists first. So protists have a highly organized nucleus and cellular organelles. Most of them are unicellular. Few are primitive multicellular organisms that have unique nuclear structures, such as a histone-independent condensed chromatin in dinoflagellates, and two structurally distinct nuclear pore complexes in ciliates. Examples of protists are algae, protozoans, and slime molds. Now, the role of protists in pharmacy. Protists can be used as one of the composition or ingredients in making a medicine. Medicines that are made of protists are usually used in treatment of high blood pressure, digestion problems, ulcers, and arthritis. In addition, protists can also be used for scientific studies. As I said before, one of the examples of protists is algae. Now, I'm going to explain the role of algae in pharmacy. Marine algae have received growing attention as sources of bioactive metabolites and considered for the pharmaceutical industry in drug development. Marine algae are potential sources of highly bioactive secondary metabolites that might represent useful leads in the development of new pharmaceutical requirements. Algae have always had the potential to be beneficial to mankind. Two of many types of algae are blue-green algae and red algae. First, cyanobacteria, which is blue-green algae, used for antibiotics and pharmacologically active compounds. Mostly the basis of these depend on the properties of algae, such as the antioxidant properties, anti-cancer activity, and antiviral properties. Second is red algae. Red algae have been used as a traditional medicine for centuries. Medical science has identified a number of complex carbohydrates with unique chemical properties. For instance, certain algae extracts have antiviral and anti-tumor properties. Red algae are frequently taken as a dietary supplement. Now, I'm going to tell a little bit more about the roles of algae as an antioxidant. The most powerful water-soluble antioxidants found in algae are polyphenols, phycobiliproteins, and vitamins. Antioxidants help in the inhibition cancer growth by causing regression of pre-malignant lesions. A study has found out that many algae species have helped in prevention of oxidative damage by the process of scavenging free radicals and active oxygen, which helps in cancer prevention. Antioxidants are the key to fight out various diseases, including chronic disorders, cardiovascular disease, and inflammations. Polyphenols, found mostly in marine algae, are having good antioxidant properties, also known as fluorotanins. The sulfates polysaccharides which are isolated from marine algae, release radical scavenging activities. Several methods of extraction are designed by researchers for this. Filamentous green algae have great antioxidant properties.
So now we will be talking about the roles and the benefits of marine algae. So marine algae, they are rich in bioactive compounds. They are considered as a potential source of bioactive substances such as proteins, lipids, and polyphenols, possessing potent antibacterial, anti-cancer, antioxidant, antifungal, and antiviral properties. Marine algae, they can also limit the development of cancers by enhancing antioxidant properties. So, antioxidants play a prominent role in later stages of cancer development. The most powerful water-soluble antioxidants found in algae are polyphenols, phycobioloproteins, and vitamins. The antioxidants may be able to cause the regression of premalignant lesions and inhibit their development into cancer. It is found that several algal species have prevented oxidative damage by scavenging free radicals and active oxygen, and hence able to prevent the occurrence of cancer. The last is as an anti-inflammatory. So marine algae, they contain polysaccharides, fatty acids, terpenes, proteins, phenolic compounds, and other bioactive phytochemicals, having the potential to be employed as anti-inflammatory therapeutics with fewer side effects. As mentioned before, example of lower eukaryotic cells are fungi and protists. Now we will be talking about roles of fungi in pharmacy. So fungi in pharmacy, the first one, they naturally produce antibiotics such as penicillin and cephalosporin. And the second one, immunosuppressant drug cyclosporin, the precursors of steroid hormones and ergot alkaloids used to stop bleeding are also made from fungi. Examples of fungi are yeast, molds, and mushrooms.
Next, I'm going to explain the substances that can be found in fungi that are beneficial in pharmacy. The first one is cephalosporin. Now, cephalosporin are a class of beta-lactam antibiotics originally derived from the fungus Acremonium, which was previously known as Cephalosporium. It was first discovered in 1945 by Italian pharmacologist Giuseppe Brotzu, which later, after the first antibiotic agent was developed from it, it was first sold in 1964 as the antibiotic agent called Cephalodin. Cephalosporins are used for the prevention and treatment of infections caused by bacteria susceptible to this particular form of antibiotic. Generally, they are active predominantly against gram-positive bacteria such as Staphylococcus and Streptococcus. Therefore, they are used mostly for skin and soft tissue infections and also the prevention of hospital-acquired surgical infections. Cephalosporins are also categorized into generations, with successive generations having increased activity against gram-negative bacteria, although often with reduced activity against gram-positive organisms. Now, cephalosporins are bactericidal, meaning it's a substance that kills bacteria. They have the same mode of action as other beta-lactam antibiotics, such as penicillins, but are less susceptible to beta-lactamase, which is the enzyme produced in bacteria that provide multi-resistance to beta-lactam antibiotics. Cephalosporins works by disrupting the synthesis of the peptidoglycan layer, forming the bacterial cell wall. Now, the peptidoglycan layer is important for the cell wall structural integrity. The final transpeptidation step in the synthesis of the peptidoglycan is facilitated by penicillin-binding proteins, or PBPs. PBPs binds to the DLA-DLA at the end of the neuropeptides, or the peptidoglycan precursor, to cross-link the peptidoglycan. Now, beta-lactam antibiotics, such as cephalosporins, works by mimicking the DLA-DLA site, thereby irreversibly inhibiting PBP cross-linking of peptidoglycan and compromising the cell wall structural integrity. Next, we have cyclosporine. Now, cyclosporin is a calcineurin inhibitor used as an immunosuppressant medication. It is a natural product, first isolated in 1971 from the fungus Tolypocadium inflatum and came into medical use in 1983. It can be taken by mouth or by injection into a vein for rheumatoid arthritis, psoriasis, Crohn's disease, nephrotic syndrome, and in organ transplants to prevent rejection. It is also used as eye drops for keratoconjunctivitis sicca or dry eyes. Now, as said, cyclosporin is used for organ transplant and other diseases that have that mainly focus on the immune system. Now, cyclosporin is approved by the FDA to treat and prevent graft versus host diseases in bone marrow transplantation and to prevent the rejection, as said, of organs in transplants. It is also approved in the US for treating rheumatoid arthritis and psoriasis, persistent nummular keratitis following adenoviral keratoconjunctivitis, and as eye drops for treating keratoconjunctivitis sicca or dry eyes caused by Sjogren's syndrome or meibomian gland dysfunction. Now, the mechanism of action of cyclosporin. Cyclosporins work mainly by lowering the activity of T cells. It does so by inhibiting calcineurin in calcineurin photophase pathway and preventing the mitochondrial permeability transition from opening. Now, the cyclosporin binds to the cytosolic protein cyclophilin, or the immunophilin of lymphocytes, especially of T cells. Now, this cyclosporin-cyclophilin complex inhibits calcineurin, which is normally responsible for activating the transcription of interleukin-2. Now, in T cells, activation of the T cell receptor normally increases intracellular calcium, which acts via calmodulin to activate calcineurin. The calcineurin then dysphosphorylates the transcription factor of NFAT, or the nuclear factor of activated T cells, which moves to the T cell nucleus and increases the transcription of genes for IL-2 and related cytokines. Cyclosporin, by preventing the dysphosphorylation of NFAT, leads to reduced effector of T cell function. Now, although it does reduce the effector of T cell function, it does not affect cytostatic activity. Pharmacokinetics-wise, cyclosporin is a cyclic peptide of 11 amino acids. It contains a single D amino acid, which is rarely encountered in nature. Also, unlike most peptides, cyclosporin is not synthesized by ribosomes. Cyclosporin is also highly metabolized in humans and animals after ingestions. The metabolites, which include cyclosporin B, C, D, E, H, and L, have less than 10% of cyclosporin's immunosuppressant activity and are associated with higher kidney toxicity. Now, individual cyclosporin metabolites have been isolated and characterized, but do not appear to be extensively studied.
That's all from us. Thank you for your attention.